Okay, so welcome to this video. In this video, what we're going to discuss is methods for investigating exocytosis. Okay, and we're going to have a specific focus on exocytosis of synaptic vesicles from axon terminals. However, for some of the experiments that we see, we won't be using axon terminals of neurons. Instead, we'll be using a very much so related cell, which is the adrenal chromatin cell. Okay, and the mechanism of exocytosis in the adrenal chromatin cells is the same as the mechanism of exocytosis in axon terminals. Okay, so let me just make sure that everyone knows what an adrenal chromatin cell is. Okay, so if I start off by drawing an adrenal gland, so remember, adrenal glands sit above the kidneys, so you have two kidneys and therefore two adrenal glands. And adrenal glands can be split into two main portions. There is the outer portion of the adrenal gland, which is known as the adrenal cortex, okay, and that contains cells which produce steroid hormones. There are three main layers in that cortex which we're not going to go into. The portion that we're interested in is this central portion of the adrenal gland, which is known as the adrenal medulla, and it's this portion that contains loads of cells which are known as adrenal chromatin cells. Okay, now these are the cells of the adrenal glands which release adrenaline into the blood. Okay, and excuse me, the adrenaline, prior to being released, is stored in little vesicles known as secretory granules. So if this is an adrenal chromatin cell, here is its nucleus. Basically, inside the cytoplasm, you have loads of little vesicles which are full of adrenaline, and these are known as secretory granules. And these are analogous to the synaptic vesicles at the axon terminal of neurons, basically. And the mechanism of exocytosis of the secretory granules, whereby they release the adrenaline within them into the blood, is the same as the mechanism of exocytosis by which synaptic vesicles are exocytosed into the synaptic cleft. Okay, right, so that's what an adrenal chromatin cell is, and certain of the experiments that we look at will be using adrenal chromatin cells rather than axon terminals of neurons. Okay, so let me outline then the structure for this video. What we're going to start off by doing is having a recap of the exocytosis process we'll specifically be looking at axon terminals, but it's the same mechanism in these adrenal chromatin cells. Okay, so that's so that we can set the scene for what we're then going to move on to. After that, we will look at certain experimental techniques that can be used to investigate exocytosis. Okay, so we'll look at in vitro fusion assays. Specifically, we'll look at lipid mixing assays and also content mixing assays. Then what we'll move on to is imaging of cells actually exocytosing vesicles, okay, using TIRFM microscopy, total internal reflection fluorescence microscopy. Okay, then finally what we'll talk about is other mechanisms, other methods for being able to measure exocytosis of vesicles. So we'll talk about capacitance measurements, and we'll also talk about amperometry, which can be used for certain neurotransmitters to measure the amount of neurotransmitter that is being released. It can particularly be used for measuring the amount of adrenaline, noradrenaline, dopamine release, the catecholamines. Okay, because there are reactions that those can undergo. They can be oxidized, basically. Okay, and we'll look at that more closely later on. Okay, so for now then, our first job is to just recap the basic exocytosis pathways so that we have a thorough understanding of that, so that when we now discuss the experiments, we actually know what we're actually trying to investigate, because there's no point talking about how we're going to investigate this thing if we don't actually know something about it prior. Okay, right, so I'm going to tell you one of the leading models for exocytosis at the moment. Now, it's important to note that the model I'm going to show you is not undisputed. It's not undisputed fact, basically. Instead, it is still controversial, but I think this is the leading model for how exocytosis works in axon terminals of neurons and also in these adrenal chromaffin cells. So I'm going to start off with the big picture of synaptic vesicle exocytosis in axon terminals, and then we'll look at it in much more detail, where we'll look at the individual snare proteins that are going to be involved in fusing the vesicle with the plasma membrane. Okay, so let's start off with the big picture then. So I'm going to draw an axon terminal of a neuron here. Okay, like so. So this is the axon ending with the axon terminal here. Now basically, in the axon terminal cytoplasm, you have vesicles which are going to be full of neurotransmitter. Okay, and these vesicles that are full of neurotransmitter, these are known as synaptic vesicles. Okay, so I'll just mark on the the neurotransmitter now. So I'll put little green dots inside them, which can represent the neurotransmitter. Okay, now, basically, what happens when an action potential arrives in the axon terminal? What happens is the axon terminal is going to release neurotransmitter into the synaptic cleft, okay, which is the space between the presynaptic cell membrane and the postsynaptic cell membrane, which I haven't drawn here. Okay, now, it's not the synaptic vesicles that are here floating around in the cytoplasm that are actually going to be released when the action potential arrives. Instead, what's going to happen is you're going to release synaptic vesicles that are actually attached to the presynaptic membrane here. So this membrane that will be facing the postsynaptic membrane, the membrane of the postsynaptic cell, this is known as the presynaptic membrane. Okay, now the portion of cytoplasm just underneath the presynaptic membrane, this sort of portion of cytoplasm, let me sort of outline it. This sort of area here in turquoise, this has a special name. This is what's known as the active zone of the axon terminal, and this is where you're going to put certain synaptic vesicles so that they can be attached to the presynaptic membrane and primed there in order so that when an action potential arrives in the axon terminal, that synaptic vesicle can then fuse with the presynaptic membrane. So this is the active zone. Okay, right. So what we need to do then in preparation for an action potential arriving in the axon terminal is we need to move some of these synaptic vesicles into the active zone so that they are ready to fuse with the plasma membrane when an action potential arrives in the axon terminal. So basically what happens is synaptic vesicles move into the active zone, and proteins in the synaptic vesicle membrane are going to interact with proteins that are at the active zone. And this process of attaching that synaptic vesicle into the active zone is what's known as docking. Okay, and this is a phenomenon that isn't actually that well understood. Okay, the phenomenon of how you bring in the synaptic vesicles into the axon terminal and then attach them there. Okay, this phenomenon isn't so well understood. Okay, the next phenomenon is the one that we're going to be really interested in, which is the priming phenomenon. Okay, so docking is just bringing synaptic vesicles into this active zone area. Okay, and we don't fully understand which proteins are involved in doing that. Okay, priming, which is the next process, is getting this synaptic vesicle that we've brought into the active zone ready so that if an action potential does arrive in the axon terminal, it's ready to actually fuse with the plasma membrane. So we are going to build complexes between proteins in the synaptic vesicle membrane and proteins in the plasma membrane, which are going to be called snare complexes. Okay, and this is going to prime this synaptic vesicle ready such that when an action potential arrives in the axon terminal, this primed synaptic vesicle is then ready to actually fuse with the plasma membrane. Okay, so that's the phenomenon that we're going to be really interested in, the formation of these snare complexes between the snares that are in the vesicular membrane and the snares that are in the plasma membrane. Okay, so priming is done by snare complexes, and there is still debate as to whether the snare complexes are the things which are involved in docking as well. Okay, maybe it's the case that priming and docking are actually the same thing. They're both mediated by the snare complexes, but we think there are probably other proteins that are involved in bringing it here prior to the actual priming process, which is the formation of the snare complexes. Okay, then after you've got these primed synaptic vesicles which are attached to the plasma membrane, which, by the way, this store of synaptic vesicles in the active zone, which are all primed, that store is now known as the readily releasable vesicle pool. Okay, or sometimes just called the readily releasable pool of vesicles. Okay, right. So now what's going to happen is when an action potential propagates along the axon, okay, so here comes an action potential, an AP, what's going to happen is it's going to make its way into the axon terminal, and now it's going to cause the fusion of the primed synaptic vesicles with the plasma membrane. Now, how does this occur? Well, basically it's going to cause the opening of voltage-gated calcium channels which are opened by depolarization. So when the action potential arrives in the axon terminal, of course, part of the action potential involves depolarization. The up phase involves depolarization. Okay, so that depolarization is going to cause the opening of these voltage-gated calcium channels here, which I'll colour in blue. And when those voltage-gated calcium channels open, you're going to get calcium moving in because of the huge concentration gradient of calcium favouring its movement into the cell. So extracellular calcium concentration is usually around 1.5 millimolar, whereas intracellular calcium concentration is usually around 100 nanomolar. So that's a 15,000-fold concentration gradient of calcium favouring its movement into the cell. So whenever you open a calcium channel in the plasma membrane, basically, you always get calcium coming in. Okay, so these voltage-gated calcium channels will open, they'll bring calcium in, and then that calcium signal is the signal which tells these primed synaptic vesicles here to actually fuse with the plasma membrane. So what will now happen is the vesicular membrane will fuse with the plasma membrane, and then what will happen is the neurotransmitter will be exocytosed into the synaptic cleft. Okay, so this is the exocytosis process. Okay, right, and something very similar occurs in the adrenal chromaffin cells. Okay, so what we're now going to discuss then is what proteins are involved in this priming process. So we're not going to discuss the docking process because that's not well understood at all. Okay, we're going to start with the priming process. How do you prime the synaptic vesicle so that it's ready for fusion? How do you form these snare complexes? Which snare proteins are involved in the formation of those snare complexes? Or even what is a snare protein will be the first question we want to ask. Okay. And then we'll look at how, when the calcium signal arrives, how that causes the development of this situation whereby the vesicle goes from just being primed to actually fusing. Okay, so we're going to look at all of that. Right, okay, so let's start off then with what actually is a snare protein. Okay, so firstly, snare, what does it stand for? Well, it stands for snap receptor, and there really is some horrible nomenclature here, so we'll go through this nice and slowly. So snare stands for snap receptor. The SNA is obviously for the first three letters of snap, and then the RE is for receptor. Okay, but then of course the question is, well, what does SNAP stand for? SNAP stands for soluble NSF attachment protein. Okay, so soluble, and then the N is for NSF, okay, then the A is for attachment, and then the P for protein. Okay, and then of course the question is, well, what does NSF stand for? And I'm afraid it doesn't end there, basically. There's another acronym within what NSF stands for. So NSF stands for NEM, okay, so that's the N, and then it's NEM sensitive factor. Okay, and then finally, once we've defined what NEM is, then it ends, basically. So NEM is the final one, basically. So what does NEM stand for? NEM stands for N-ethyl melamide, okay, which is a chemical, okay, with a very beautiful structure actually. Right, so that is what a snare actually is, or at least what the nomenclature stands for. It stands for SNAP receptor, okay? So, but what actually is a snare protein then? What is the feature that all snare proteins share? Well, the feature that all snare proteins share is that they have a very long alpha helice of around 50 to 60 amino acids. So the key feature then that snare proteins all share is that they have a long alpha helix, like so, consisting of between 50 and 60 amino acids. So this will be between 50 and 60 amino acids. And this long alpha helix, which is a spring-like structure where the polypeptide folds up into this spring-like shape, this is known as the snare motif. Okay, so all snare proteins have a snare motif which is between 50 and 60 amino acids in length. Okay, right. And what's going to happen basically is we are going to have snare proteins on the vesicular membrane. Okay, so to draw a picture of this, we're going to have our vesicle here, and we're going to have our plasma membrane here, and we're going to have snare proteins that are within the vesicle, which will be known as V-snares. Okay, and I'll just bring this up a little bit. Okay, so V-snares standing for vesicular snares. Okay. And we're also going to have snare proteins which will be in the plasma membrane, which will be called T-snares. And the T is for target snares here, so T-snares. Okay, so this T is for target. And the idea basically is that the alpha helices, the snare motifs of these snare proteins, are going to intertwine with one another and form a bundle. In fact, what's going to happen is you're going to have, in total, 4 separate snare motifs, and these snare motifs are all going to bundle up and form what's known as a 4-helix bundle. So let me try and show you what this means. Okay, so basically the idea is that these snare motifs, I'll draw one here, are going to intertwine to form this 4-helix bundle, like so. So here's one snare motif, and then we'll have another snare motif intertwining with this one like so. Okay, and then we might have another one intertwining with this one like so. Okay, and then finally another one on top of that, so that you've got overall 4 of the things intertwining together to make this 4-helix bundle, as it's called. And the idea is that these V-snares and the T-snares, the snare proteins that are within the vesicle and the plasma membrane respectively, are going to intertwine together to form these 4-helix bundles, which are going to be attaching these 2 membranes together basically, and that's going to be a very important part of the priming of synaptic vesicles for fusion. Okay, right. Now, what I should say is that there are 4 different types of snare motifs, okay? And when we form these 4-helix bundles, it's going to involve one of each type of the snare motifs, okay? So you're going to put 4 snare motifs together to make the 4-helix bundle, and you're going to have one snare motif from each type, basically. So what are the 4 types of snare motifs? Basically, you have the R snare motifs, QA snare motifs, QB snare motifs, and also QC snare motifs. So these are the 4 different types of snare motifs, basically. When we form these 4-helix bundles, it's going to involve one of each type of snare motif. Okay, right. So now what we're going to talk about is the specific types of snare proteins that are going to be involved in the formation of these snare complexes in synaptic vesicles. Okay, so we're going to talk about specific examples of snare proteins. So the first one that I'm going to talk about is syntaxin 1. Okay, so this is going to be a T-snare. It's going to be in the plasma membrane, and it's got a pretty interesting story about how it actually gets into the active zone and how it actually gets ready to interact with other snare proteins to form this 4-helix bundle. So let's start with syntaxin 1 then. Okay, so Here then is the plasma membrane, okay, and I'm drawing the plasma membrane as two parallel lines. One of these lines represents the inner leaflet, and in this case it's going to be the top line that represents the inner leaflet, and the other is going to represent the outer leaflet, and that will be the bottom line here. So this side above here is going to be the cytoplasmic side, and the side below here is going to be the extracellular fluid side, the ECF side. Okay, so syntaxin 1 is going to be an integral membrane protein bound by one membrane-spanning alpha helix into the plasma membrane here. Okay, like so. So here, this polypeptide is representing the protein, and what's going to happen is the C-terminus is going to be on the extracellular fluid side, and then on the cytoplasmic side you're going to have its snare motif, like so, and then after the snare motif you're going to have a region known as the HABC domain, which consists of 3 alpha helices. Right, so I'm going to colour in different bits of here to show where the alpha helical structures are. So this portion here in turquoise, this is the transmembrane region. Okay, this is the membrane-spanning alpha helix. Now, I haven't shown it as an alpha helix, but I'm colouring it in to show that that's an alpha helix. And TMR here, this stands for trans, that's the T, and then membrane, that's the M, and then R, that's the region. Okay, so transmembrane region. Okay, then, on the cytoplasmic side, here in red, this is the snare motif, the key motif to syntaxin 1's function, okay? And for syntaxin 1, the snare motif is a QA snare motif. So this is a QA snare motif here in red, okay? Then, after the QA snare motif, you then have this cluster of 3 alpha helices together here in orange. Okay, that are all folded back against one another, and that cluster of 3 alpha helices there, that is known as the HABC domain, because it's got 3 helices together making up that little domain. Okay, right, so that's the structure then of syntaxin 1. We've seen its snare motif here, this QA snare motif. Now the important thing to say about syntaxin 1 is that syntaxin 1 actually has 2 conformations. Okay, the conformation that I have drawn it in here is currently what's known as the open conformation of syntaxin 1, but it also has a closed conformation. So let me now show you the closed conformation of syntaxin 1. Okay, so once again, here is the plasma membrane. Okay, again we'll have the C-terminus extracellularly, so here's the C-terminus of syntaxin 1. Then here is the QA snare motif, and in the closed conformation what's going to be different is that that HABC domain of 3 alpha helices sitting alongside one another is going to be folded back against the QA snare motif. So here in red, here is the QA snare motif. Okay, here in orange, this is now the HABC domain, and you can see that it's now folded back on top of the QA snare motif, and it's effectively blocking that QA snare motif from actually interacting with other snare motifs. Remember, the key thing about snare motifs is that they're going to interact with other snare motifs and form this 4 helix bundle. Okay, but while syntaxin 1 has that HABC domain bent back on top of its snare motif, and that snare motif cannot now interact with other snare motifs. So this is the closed conformation of syntaxin 1. And in fact, there is an important protein which binds to syntaxin 1 and stabilizes it in this closed conformation. Okay, and I'm going to show you this protein here now. Okay, so here it is bound around this sort of portion of the syntaxin 1 where you've got this HABC domain bent back against the QA snare motif. And this protein that binds to syntaxin 1 in this sort of conformation and stabilizes it in that conformation, that protein is known as MUNC18. Okay, so this is MUNC18 in purple here. Okay, so MUNC18 is actually absolutely essential for exocytosis in synaptic terminals. Okay, if you knock MUNC18 out of neurons, then they no longer have any synaptic vesicle exocytosis, so its function is absolutely essential. Okay, so what does the function of MUNC18, well, how do we reconcile this with MUNC18 locking syntaxin 1 in this closed conformation? Because syntaxin 1 needs to be in the open conformation to prime synaptic vesicles to form the snare complexes. So it seems as though it's a little bit of a odd thing here, that MUNC18 seems to be against exocytosis by locking syntaxin 1 in the closed conformation, but then actually if you knock it out, you get absolutely no exocytosis. Okay, well what we think MUNC18 does is it binds to the syntaxin 1 when the syntaxin 1 has been made. And basically the idea is that it helps the syntaxin 1 get to the active zone of the axon terminal, okay, without binding to other snare proteins before it actually gets to the right place. So the idea is it locks the syntaxin 1 in the closed conformation until the syntaxin 1 is actually in the plasma membrane at the active zone of an axon terminal, and then the idea is that proteins in the active zone are going to then facilitate the unloading of MUNC18 from the syntaxin 1. So the idea is that if you knock MUNC18 out, you don't get any syntaxin 1 getting to the active zone, basically, and therefore you don't get any exocytosis because syntaxin 1 is utterly essential. Okay, right. So the idea then now is that when you get to the active zone, okay, so now this piece of plasma membrane here, this is now a portion of the active zone plasma membrane. So if I draw my axon terminal again, it's a portion of this plasma membrane here in purple. This is the active zone plasma membrane, the presynaptic membrane. Okay, so let's say our syntaxin 1 has now arrived here, and it's still locked in place by the MUNC18. The idea now is that other proteins are going to bind to the MUNC18 that are in the active zone, and are going to facilitate the MUNC18 now changing its conformation, changing the way that it binds to syntaxin 1, so that it now binds to syntaxin 1 in the open state. So basically the idea is here that the syntaxin 1 is now going to move from being in the closed state to being in the open state. Potentially the MUNC18 is still bound, but now it prefers to bind in the syntaxin 1 when it's in the open state. And this is because new proteins are bound to the MUNC18. So let me just colour bits in here and then I'll explain this in more detail. So once again, here is the transmembrane region of syntaxin 1. Here is the QA snare motif of syntaxin 1. Here in orange is the HABC domain of syntaxin 1. Okay, and then here in vivid purple, this is MUNC18 here. Okay, so that's MUNC18 there. Okay, right. And now the idea is that now the MUNC18 is bound to the syntaxin 1 in the open conformation, a conformation that is able to form snare complexes. Okay, now why has the MUNC18 suddenly gone from preferring to bind to syntaxin 1 in the closed conformation to preferring to bind it in the open conformation? Well, it's these new proteins that are bound here. Now, one of these proteins that binds here is believed to be the protein MUNC13. Okay, and again, MUNC13 is a totally essential protein. If you knock it out, you get no exocytosis. Okay, and its role is believed to be the facilitation of the unloading of the syntaxin 1 effectively, the changing of the way that MUNC18 interacts with syntaxin 1 so that once the syntaxin 1 arrives in the active zone, it can be opened up basically. And then there is the potential too for other as yet unidentified active zone proteins to be binding here. So in green here, this is another active zone protein, or potentially more than one active zone protein, okay, that hasn't yet been identified. So let me summarize what the idea then here is. Basically, when the syntaxin 1 is initially produced, MUNC18 binds to it and ensures that it stays in this closed conformation. In the closed conformation, it can't interact with other snare proteins, and then the idea is that this MUNC18 facilitates the delivery of the syntaxin 1 safely to the active zone and makes sure that it doesn't interact with any other snare proteins before it gets to the active zone. Okay, then once you get into the active zone, other active zone proteins and also MUNC13, which is also in the active zone, bind to the MUNC18 and facilitate this change in conformation, so that now either the MUNC18 fully cleaves off, and this is still where we don't know what happens, either the MUNC18 fully cleaves off and then the syntaxin 1 goes into the open conformation, or the MUNC18 remains bound but it binds now to a syntaxin 1 that's in the open conformation. It just binds now to the HABC domain down here, and that's because these other proteins are bound to it. Okay, so either these proteins remain bound or they might cleave off. Now, because it will make the picture easier, what we're going to do is we're going to imagine that they cleave off, okay, but potentially they are still bound as they are in this picture. Okay, this is a part where we don't know the answer, basically. We don't know whether those remain bound for the formation of the snare complexes, or whether they cleave off and leave the syntaxin 1 in this open conformation like so. Okay, right, but for future pictures now, we're going to abandon the MUNC18, the MUNC13, and the other active zone proteins. Okay, and we've now got syntaxin 1 in the open conformation at the presynaptic membrane. Okay, right, so syntaxin 1 is one of the T-snares, okay, but there is another T-snare, so let me introduce the other T-snare, and then we'll see how the two T-snares can form a T-snare complex, and then what will happen is the single V-snare protein will then come and join this T-snare complex. Okay, right, so the next T-snare then is what's known as SNAP25. Now, what does SNAP25 stand for? Well, the big thing to stress here is that it does not stand for soluble NSF attachment protein. It actually, unfortunately, has absolutely no relation to the SNAP proteins that we were talking about when we were talking about the name of snare proteins. Okay, in fact, in this video, we're not actually going to see the SNAP proteins and the NSF proteins. We're not going to go that far. They are involved after exocytosis has occurred in breaking apart cis snare complexes. Okay, so once the exocytosis has occurred, these snare complexes end up all in one membrane basically, and breaking those apart is the job of SNAP and NSF, but we're not going to go that far. Okay, so we're not actually going to see real SNAP proteins. We're going to see SNAP25, which is called that for a different reason basically, and it has no relation to those other SNAPs. Okay, so what does this SNAP stand for then? It stands for synaptosome, that's the SN, okay, synaptosome, and then it's associated protein, so the A is for associated, and the P is then for protein, and then it's the synaptosome associated protein of 25 kilodaltons, okay, so of 25 kDa. Okay, now, just to make sure you know what a dalton is the unit of molecular weight, basically. So 1 dalton is around the weight of a proton or a neutron. Okay, so 25 kilodaltons means that this protein has the weight of around 25,000 protons. Okay, so you've added up all of the atoms that constitute it and all of their molecular weights, or atomic weights rather, and that's giving you the overall weight of this protein, and it's around 25 kilodaltons. Okay, so that's what SNAP25 stands for, not soluble NSF attachment protein. Okay, right, so let me show you the structure of SNAP25. Now, SNAP25, again, it is attached to the plasma membrane in the active zone, but it is not an integral membrane protein this time. So once again, let me represent the presynaptic membrane as these two parallel lines, and once again let the top line here be the intracellular leaflet and the bottom line be the extracellular leaflet. Okay, now, SNAP25 proteins are not going to be integral membrane proteins. Instead, they are going to be attached to the plasma membrane by lipid moieties that have been attached onto them. Okay, right, so let me just draw the picture and then I'll explain it. So this portion here, this curvy line, this represents the actual polypeptide of SNAP25, whereas these two little lines here, which I'll colour in turquoise, these represent the lipid moieties that have been attached. Okay, so these are lipid groups. So the idea here is that the protein has had lipid molecules attached onto it, okay, and these lipid molecules implant into the inner leaflet of the phospholipid bilayer and therefore attach the protein to the inner leaflet of the phospholipid bilayer. There are many proteins that are attached to the cytoplasmic face of the plasma membrane by having lipid moieties attached onto them, which then implant into the phospholipid bilayer. Okay, so that's how SNAP25 gets attached to the plasma membrane. Now, SNAP25 actually has two snare motifs, okay, which I'll colour in green here. Now, one of these snare motifs is an example of a QB snare motif, and that's the one at the amino side, and the other is an example of a QC snare motif, which is the one at the carboxylic acid side of the protein. Okay, right, so this is the other T-snare, this is the other snare protein that is at the plasma membrane. Now, the first thing that's going to happen is the syntaxin 1 that is now in the open conformation is going to form a complex with the SNAP25. Okay, so these 3 alpha helices, the 2 from SNAP25 and the 1 from syntaxin 1, are going to form a bundle, basically. They're going to intertwine before you ever get the involvement of the V-snare from the synaptic vesicles. Okay, so let me get another piece of paper and then we'll draw this. Okay, so this structure then is what's known as a T-snare complex, okay, because it's formed by the target snare proteins that are in the plasma membrane. So let's now draw the structure of one of these T-snare complexes. Okay, right, so here then, this is the plasma membrane once again, okay. Then we'll have our syntaxin 1 protein here, so here's its HABC domain, and I'm going to drop, as I said before, I'm going to just drop drawing the MUNC18 and the MUNC13 and the other active zone proteins. I'm just going to draw syntaxin 1 in the open conformation. Those proteins, however, might still be attached to this even whilst it's forming these complexes. Okay, but it'll make the picture simpler not to have them there. Okay, so here is our syntaxin 1 protein, okay, and then what we've got here is SNAP25 drawn here. So here is the amino terminus, here is the C terminus, and then you have lipid moieties attached to that protein that are attaching it into the cytoplasmic face of the plasma membrane. And here now are the QB and the QC snare motifs of SNAP25. Now I'm just going to draw these snare motifs parallel to one another rather than showing them intertwined. It makes the picture look simpler. Okay, so there's our T-snare complex. Now what we need to introduce is the V-snare. We need now to see the snare protein that is within the synaptic vesicles membranes. Okay, so we said that when we form these 4 helix bundles, it involves 4 snare motifs. We've got 3 snare motifs, so that means that the final snare protein that's going to be provided by the synaptic vesicle membrane only needs to contain a single snare motif. So let's draw our synaptic vesicle here. Okay, so here is the lipid bilayer of the synaptic vesicle shown here. And basically the snare protein that is implanted into the synaptic vesicle membrane is a protein known as synaptobrevin 2. Okay, and it has its C terminus inside the synaptic vesicle lumen and its amino terminus outside the synaptic vesicle, so in the cytoplasm. Okay, and here then is the snare motif of synaptobrevin 2. Okay, and this is going to be an R snare motif. Okay, so this protein that you're going to find within the synaptic vesicle membranes, and I should stress they're going to have far more than one. In fact, they usually have around 70 synaptobrevin 2 proteins. Oh, it's smudged horribly. Okay, in their membrane. Okay, so this is synaptobrevin 2. Now, synaptobrevin 2 also has another name that's quite commonplace to see. Okay, and it's also called VAMP2. Okay. So what does VAMP stand for? It stands for vesicle, that's the V, associated, that's the A, and then membrane protein, that's the P, the M and the P. Okay, so vesicle-associated membrane protein, and then obviously vesicle-associated membrane protein 2. So it is specifically synaptobrevin 2 that is within these synaptic vesicles. Often people will drop the 2 and they'll assume that you know what you're talking about, so they'll just call it synaptobrevin. Okay, but it is specifically synaptobrevin 2 if you want to be precise. Okay, right. This snare protein then, this is the final one of the 4 helix bundle, and this is going to come and associate with these snare motifs here to form the full snare complex. Okay, now, not just one of these snare complexes is going to form. Okay, I said that in these synaptic vesicles you have loads of synaptobrevin 2 proteins. In the plasma membrane, you're going to have loads of these T-snare complexes, so more than one of these full snare complexes can form between the synaptic vesicle membrane and the plasma membrane. Okay, in my next picture, I'm going to go from showing one of these snare complexes to showing 2. It makes the picture look beautifully symmetric, so we're going to show 2. Okay, but it could be more than 2 that actually form. Okay, so let's draw this picture, and this is the picture that we've been building up to now. Okay, so here is the synaptic vesicle membrane, like so. Okay, and now in the synaptic vesicle membrane we're going to have the V-snare, which is synaptobrevin 2 here. Okay, and as I said, we're going to show 2 of these snare complexes forming, so I'll show 2 synaptobrevin 2 molecules. So here is the R snare motif of synaptobrevin 2. Okay, now let me put in the plasma membrane here. So this is the plasma membrane. Okay, and in the plasma membrane you're going to have this syntaxin 1 and also the SNAP25 attached. So I'll start by drawing syntaxin 1. So here's one syntaxin 1, and then on the other side here you've got the other syntaxin 1. Like so. Okay, and then the final snare protein that we need to add in is the SNAP25s, which I'll put here, which will be attached to the plasma membrane by their lipid moieties here. Okay, and here's the amino and carboxy termini of these SNAP25 proteins. Okay, right, so let's colour this in. Here's the QB snare motif of SNAP25, here's the QC snare motif of SNAP25, again, for this one over here. Okay, colouring in the portions of syntaxin 1. Here is the QA snare motif of syntaxin 1. Here is it for that second syntaxin 1. Okay, then we have the transmembrane region of syntaxin 1, and then we'll also mark on the HABC domain here in orange. Okay, like so. So, this complex that we have now got formed here, this is what's known as a trans-snare complex, and in fact we're showing 2 trans-snare complexes here. So a single trans-snare complex is the synaptobrevin 2 snare motif with the SNAP25 snare motifs and the syntaxin 1 snare motif. So this is a trans-snare complex. Now why is it called a trans-snare complex? Well, the snare complex hopefully makes complete sense, but the trans, what does that mean? Trans means on the opposite side to. Okay, so this is referring to the fact that the snare proteins are in opposite membranes that are involved in the formation of this snare complex. Okay, right, so there now we have got the formation of 2 trans-snare complexes shown. Now, let's talk about this a little bit more. Basically, when you form these trans-snare complexes, this 4-helix bundle, which is the crucial concept here, this 4-helix bundle, which is the concept of these 4 snare motifs intertwining up together and winding around one another. Basically, this winding round process begins at the amino terminal ends of the snare motifs, or at least at these ends. Of course, this is the C-terminal end of this QB snare motif, but it's beginning at this side over here, which is usually the amino terminal end of the snare motifs. At least it is for this one, this one, this one. It's not obviously for this one, but it's this end, basically. So the zippering begins here, and then works its way back, and zippering is the key word that people use to describe this. Okay, the snare motifs zipper up. okay, and it propagates backwards like so. Now, when this occurs, it releases a lot of energy, okay, and it's this energy that is used to pull the two membranes closer together. Now, why do you need to put energy in to pull the two membranes closer together? Well, phospholipids, which are the things out of which phospholipid bilayers are produced, have polar heads, and these polar heads are generally negatively charged. So basically, this membrane here is negatively charged. This membrane surface here is negatively charged. You're trying to bring two negatively charged structures together. That's why you have to put in energy. And it's the energy released by the formation of these four helice bundles that is going to pull those two membranes together. Now, there's a problem here, which is that if you just let this roll, what seems to happen is that it would actually just fuse these two membranes together. Now that's not good, okay, because that means as soon as the vesicle arrives and primes, you're just going to get fusion, okay, and that's not going to link fusion of the vesicles to the arrival of an action potential. So you don't just want to be releasing vesicles randomly, you want them to be released in response to an action potential arriving in the axon terminal. So something has to stop the progression of the zippering up process, to stop it getting right to the end, because if it gets to the end, these two membranes are going to be pulled so close together that they fuse. Okay, so what happens, or at least what is believed to happen, and again it's a good moment for me to stress that this is the leading model for how this works, but it's not undisputed. Okay, so the leading model says that what happens is another protein now binds to this trans-core snare complex, okay, and blocks the progression of the zippering up to stop it completing, okay, and this protein is a protein known as complexin. So I'll put one on each of these trans-snare complexes. So this protein here, this is complexin, okay, right, and I will underline complexin in yellow here. So the idea then is a complexin protein associates with each trans-snare complex, and basically the idea is that it clamps that trans-snare complex and stops the progression of the zippering up. So complexin is often described as a clamp protein, okay, and this theory of how complexin works is often called the clamp theory. Okay, so it binds to the trans-snare complex and stops the progression of the zippering up. So let's say it halts it here, and then you have the membranes very closely together, but they're not close enough to fuse yet. And the idea is that that then is the finished primed vesicle. Now the vesicle will sit in that position and wait for an action potential to arrive, and only when the action potential arrives is this clamp protein, this complexin, removed, and then the zippering process can continue on, it can finish, and it can bring these membranes so close together that they then fuse. And that's then how we link the fusion process to the arrival of an action potential in the synaptic terminal. Okay, so now then let's talk about how you sense the arrival of the action potential, and then how you remove the complexin protein to unleash the clamp to allow this fusion to actually occur in response to that action potential. Okay, so let me just remind you of the basics then that I talked about earlier. Okay, so once again, here is our axon terminal. So let's say an action potential is propagating down this axon into the axon terminal. We know that the key thing that occurs is calcium signaling. The electrical signal of the action potential is converted into a calcium signal, okay, by voltage-gated calcium channels. Okay, so here is our voltage-gated calcium channel here, VGCC for short. Okay, and when the action potential arrives here, you're going to get depolarization of the electrical potential difference across the cell membrane. That's going to trigger the voltage-gated calcium channel to open. It's going to allow calcium from the extracellular fluid to come into the cytoplasm, and this calcium is now the trigger. Okay, this is the trigger for this complexin to be removed. Okay, but it's slightly more complicated than that, because it's not just the case that calcium binds to the complexin and it cleaves off. Complexin doesn't have a calcium binding site. So it works for another intermediate protein. Okay, and it's this other protein that we now need to talk about, this calcium sensor. Okay, and this important calcium sensing protein is what's known as synaptotagmin. Okay, right. So, I think we will call it there for this video. We'll leave on a cliffhanger. And in the next video, what we will do is we'll continue this discussion. We'll look at the structure of synaptotagmin. We'll look at how, when the calcium signal arrives in the cytoplasm of the axon terminal, synaptotagmin is going to behave after that in order to remove the complexin protein from these trans snare complexes and trigger the completion of the fusion event.